My name is Mathew Schwartz, and I'm an assistant professor at the New Jersey Institute of Technology. In this video, I'll be presenting the paper Automated Multi-Feature Segmentation of Treadmill Running. This presentation will focus on explaining the algorithm and methodology used in the research. The goal of our work is to take the marker trajectories from motion capture data of a given movement and extract the same or most similar movement. The purpose of this extraction is to enable the comparison between the same motion of a subject or amongst multiple subjects. There are numerous papers focusing on the analysis of time series data and, more relevantly, on event detection and labeling. The problem in relation to the movement sciences is usually formulated around the illustrated video. Given a series of marker trajectories, how do we determine exactly which frame are the start and end of a specific movement? As the video shows, a common practice is to step frame by frame and determine a reasonable location for an event. In this case, the z-axis in the top graph is expanded to illustrate the difficulty in making a decision in the case of a toe-off event. The approach in this paper was motivated by the combination of multiple factors. First, common methods for event segmentation or labeling rely on force plates, which may or may not exist. Likewise, there are often usable movements that occur before and after the force plate in a lab. Second, long motion trials that may not have been the focus of a movement can still be useful. For example, trials that recorded someone stepping over an object may also have useful regular gait data. Third, manual labeling is time-consuming and inconsistent. Using an automated process can improve consistency. Finally, the aim is not to rely on any particular feature, such as defining rules about a specific marker velocity. Similarly, the method should not rely on learning from a large dataset, requiring training whenever a new segment of motion is desired. Essentially, the problem is formulated as, given a sequence of data, how can we extract subsequences of interest? There are three main phases of the segmentation algorithm. First, the data is processed for features. Next, these data sets are compared with a rolling correlation, and last, the aggregated correlations are clustered to determine the best candidates. In the following slides, I'll explain these steps in more detail. For each frame in the trial, we reorient the pelvis direction along a single axis so that variations in walking direction across different trials are normalized. At this step, additional features are also created, such as the distance between markers. Multiple references of the same motion get resampled to match different time series. Features are correlated amongst the reference set, and we store the importance of each feature. For the second phase, a few parameters are set, such as the scales to use for the rolling window. Then, for each of the features that exist in both reference and target data, we iterate through the reference set for each scale in the set. The correlation between each of the references to the target data is calculated across the entire trial. The value of the correlation for each timestamp is stored and the correlations are aggregated, in this case by summation. Important to note, this is a rolling window and not cross-correlation. The final phase of the process is extracting the desired segments. Here is a sample visualization of multiple features correlated over time. The features are averaged for each time step, and using a peak detection algorithm, the most correlated regions are found. For the given peaks at the reference scale, a portion of the timeline is reserved for the start of the peak and the length of the reference. This is repeated for each scale. During this process, subsequent and lower scoring blocks are checked to be overlapping with an existing one. A new block is created only if there are no existing segments. Following this process, a time series is blocked out with the desired segments. However, some segments may be unique, but also not the desired movement. A final clustering step is used to determine if there are any block segments that are significantly lower in similarity than the group of segments found. The method presented was applied to 16 subjects of running with 2 to 3 full strides each. 6 instances for right foot strike and right foot off were used as reference data to find the same segment in the other subject trials. The image on the right shows the distribution of the 37 start and 37 end timestamps determined by the algorithm and compared to the original human labels. As a visual reference, this example shows the output of the algorithm on the right, extracted from the full running sequence on the left. The algorithm found 3 segments of a right foot off to right foot on sequence in the full running trial on the left. The segmentation portion of this algorithm is available on GitHub. As a note, it does not include the motion capture data, loading, or feature processing. Thank you, and if you have any questions, please email me or feel free to open an issue on the GitHub repository if you would like to apply this to your own dataset.